Good morning, students. Good morning, students. Am I audible? Any response? Am I audible? Can you hear me?
Yes, ma'am. Good morning, ma'am.
Good morning. At least one student has responded. Okay. Before I start, let me see how many of you have joined. Only 13 students. Wow. What a great thing. Before I start, just give me a review that is how I was teaching yesterday using the digital graphic board. Is that comfortable for you? Are you guys comfortable with that? Or should I start using Blackboard again?
Ma'am, Blackboard.
See, Zoom thing is not in my hand because that is complete. Okay. Yeah, I understand. But the classes I have to take anyways in Zoom because na ID things are there. Okay, that thing you. Okay, okay, I'll do that. So you are saying that I should just change in the software, but how many of you think that I should go back to whiteboard? How many? Just raise your hand. How many of you think that? Show Mando, only two participants out of this 17, three participants. Anurag, Achanisha, Anurag, Anisha, Anurag, Shomalo, three. Yesterday I got one review. So fine, that's a different thing. But one informal announcement I'm doing now. Your school may restart from 3rd of February. Not may, your school will restart from 3rd of February. So get your dresses ready, get your school bags ready because
Bus will give. Bus.
most probably bus and transportation will be given. See that thing again not in my hand because I'm not handling that. The announcement we came to know as per that the school will restart soon. So that's a big thing.
Yesterday only we got the news.
Yesterday only, government has given a green signal for reopening of school. So that thing we will do. We are working on that. So yeah, so digital board, whiteboard, this problem is no longer going to exist because anyway we are going to start physical classes, and it's a great opportunity for this is a great opportunity for me, and I don't know about you people because যত রাজ্যের ফাঁকি মারার ধরো মেরে ফেলেছি, now it's our time. Now we will be handling you people well. But I want to have full attendance of my PCMB students. Get it be very cleared that PCMB students should be present every day. If the thing is the transportation is being provided, then the very good. else you try to come. Those who are coming at least, like Anisha, Apurupa, Shomallo, Diptangshu, Shukriti, Shuprakash, Aditya, or Joita. These people I want every day in the branch so that you can attend the classes as per your routine. And this time it's a very big thing. This time it's a very big news. Okay, so you people please join and like এই join কথাটাই বন্ধ হয়ে যাবে, attend the classes. Where is Shukriti? Shukriti is not there. Diptangshu is there.
Well, transport will not be provided.
Transport will be provided as per my understanding that transport will be provided. Acha, from yesterday's part, anyone having any doubt? From yesterday's part, anyone having any doubt? First, tell me that, then I'll move to the new portion. Anyone having any doubt in the last part?
No, ma'am.
Somal, are you sleeping?
No, ma'am.
Eating then?
Yes.
Okay. Just one thing. So if you people are not having any doubt, so just tell me, let me ask some question and you people try to give answer of those questions. Okay. Let's start with Apurva. Tell me now, just give me an idea that what are there inside our muscle. If we cut our muscle and see the inside of our muscle, what will we see? What we will find? Tell me. What happened? Apurva, am I not audible enough? Anurag,
Yes, ma'am.
tell me. If we cut our muscle and see the inside of our muscle, what we will see? What we will find?
Ma'am, biceps, ligaments, and tendons.
Biceps, ligaments, and tendons. That is right. But if we cut the muscle, bicep, then if we cut the bicep, then what we will find? If we cut our biceps, then what we will find?
How can I try?
Ma'am, myofiber muscle cells.
New fiber muscle cells. Acha, Nikita, yes, you can try.
If we cut a muscle cell, then we find the outer layer, which is epimysium. Inside that, there are small pipe-like structure, which is known as fascicle. And inside fascicle, there is small cigarette-like structure, which is known as myofiber.
Okay, and then keep
And
digging
inside myofiber, there is myofibril.
Okay, just then I'm trying to draw and just tell me that what are the names of the parts. One by one, you just tell me what are the names of the part. So, this is a skeletal muscle. This is skeletal muscle. Okay, and now what we have done, we have cut this part of the skeletal muscle. Okay, after cutting, I have turned this part towards you, so we can see around this type structures. Okay, what? So this is a muscle. First we will get this round structures. What does this thing called?
Fascicle.
Fascicles. Fascicles, right. So this layer of the muscle is called outer layer. What does this thing called?
Epimysium.
Epimysium, right. After epimysium, what does this thing called? The outer layer of the fascicles?
Perimysium.
Perimysium, right. Now, if we dig deeper, then we will find that, but we will find that here is again some round structures. Let's make it 3D. Again, inside of the fascicles, some structures are present. What does this thing called?
Myofiber.
Myofiber. Again, inside of myofiber, again some cigarette-like structures are found, which are called as
Myofibril.
inside the myofibril. So inside of the myofiber, again some cigarette-like structures are found, which is called as myofibril. Varun, are you listening? Varun? Oh, Varun.
Yes, ma'am.
Okay, are you listening?
Yes, I'm listening.
So, this is myofibril. One by one, those who have missed yesterday's class because it was in 4 p.m., it was your sleeping time. So, for them only, I am recapitulating this part, that what is this is if we cut the muscle, this part I have cut and turned it towards you, so we will see a round like structure. This structure will be visible to us. The outer layer is called as epimysium. Then the middle layer of the fascicle covering, the covering of the fascicles are known as perimysium. Perimysium. And lastly, perimysium. Lastly, this part, the part which is in which is so the covering, this covering of the entire myofiber is called as myofibers covering is called as endomysium. Okay, this is a myofiber, and which is also the muscle cells. Myofibers are also called as the muscle cells. Lastly, this is myofibrils. Myofibrils. Myofibrils, right? Now, if you have understood up to this, just tell me, what does this structure called? You will only tell, I'll write. What does this structure called? This roundish structure? Fasci? Fascicle. Fascicles. Fascicles. Shrishant, are you listening? Fascicles. Acha. Why I have recapitulated entire thing? Because yesterday I have not given you one information about all these fascicles are not hanging. they are not hanging. Rather, they are connected with the help of some uh, connective fibers. This in these open parts, some connective fibers are present which help these fascicles to be in a certain position. All these fascicles are definitely not hanging in the myofiber or muscle, right? So they are connected with some connective fiber, which is this red colored fiber. Okay, this is having a name. This is having a particular name. This red colored thing, this red colored fiber is this part is called as fascia. Fascia. This red color thing is called as fascia. 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 Okay. To this part, this should be completed. Now, if you remember, we had a close discussion of only this part. That is this part. We have cut a portion of this part. Suppose we have cut a small portion from this part, and then we had a detailed discussion over of that. So I'm drawing it here for just a quick recapitulation. This is the myofiber, or which is also called as muscle cells. This is the structure of a muscle cell. This is the structure of a muscle cell. The infoldings which I have drawn inside the muscle cells are called as, what does this things called? These infoldings are called as,
T-tubule.
yeah, it is called as T-tubule. For tomorrow's homework, you have to tell me what is T-tubule, what is the function of T-tubule. If you can't, then I'll definitely tell, but first you try. So inside, if it is a myofiber, if it is a structure of a myofiber, then the structure inside of the myofibers, the cigarette like structures inside a myofiber is called as this
Myofibril.
why only Nikita is giving answer? Rest of you are sleeping. Is it not morning enough for you? Okay, so these are myofibrils. Myofibrils. So, what is the plasma membrane of myofiber called? What is the plasma? Anurag, what is the plasma membrane of myofiber? Yes, Anisha.
And sarcoplasm.
Plasma membrane.
Sarcolemma.
Sarcolemma. Yes. Sarcolemma. So what is then sarcoplasm? As Anisha told sarcoplasm, what is sarcoplasm? What is
Cytoplasm of myofibril.
So cytoplasm of myofibril, this is the cytoplasm which I am trying to draw with yellow color. This is the cytoplasm of myofiber, which is also called as sarcoplasm. Acha. Now tell me, it is uninucleated or multinucleated?
Multinucleated.
Okay. Alicia, are you there? Alicia, are you there? Why no response I am getting from you? So it is multinucleated. Now you tell me, if one endoplasmic reticulum is present, suppose this is an endoplasmic reticulum, what does it will call?
Sarcoplasmic reticulum.
Sarcoplasmic reticulum. Lastly, anything I'm missing? Sarcoplasmic reticulum is done, sarcoplasm is done, plasma membrane is clump. Two things I have not told you yesterday, maybe I have missed. First thing is that it contains glucose. Muscle contains glucose. Suppose this is a glucose molecule. This is again a glucose molecule or glycogen molecule. Can you tell me why muscle contains glucose? Why muscle contains glucose? Because you never know what emergency can happen when you have to run, when you have to fight, when your muscles need to contract, or how. Suppose you are coming to school from day after tomorrow, and suddenly one mad dog is running for you. That time you have to run like anything, so that time you need glucose in your muscles. So what muscle does? Now muscle store its own glucose. So it store its own glucose, which is known as muscle glycogen. Muscle glycogen. Muscle glycogen. Okay, so it is called as muscle glycogen. Again, not only, what about, we all know that glucose plus O2 is equal to ATP. So now glucose is here, glucose is present here as my muscle glycogen. What about oxygen? If oxygen is not present, then it is also not possible to make ATP and run. So muscle fiber have their own pigments, okay? Like our body, our blood is having its own pigment which is known as hemoglobin, which contains and carry oxygen. Muscle fiber or muscle fibers or myofibers also carry their own pigments that contain oxygen. So instead of drawing in a new picture, yesterday's drawing you have, if you have done, then yesterday's drawing, if you have done, just add these two features, glycogen part and the pigment part which I am drawing. These two part you add in that picture. So what does this pigments call? What does this red colors pigment call, which is found in muscles? They are called as this red color. This red colors pigment are called as myoglobin. Just like hemoglobin, it is called as myoglobin. GLOBIN, myoglobin. Okay, myoglobin. Clear? Everybody clear? Myoglobin. Do it fast. Next, we will move. Acha. Yesterday, I have told you one more thing, that is, it is having this microfibrils are having small. They are having very small banding in them. So now today we will read what are the reason of this bandings, what are the reason of this bandings. Okay, Can you tell me what are the reasons of this banding? Because I know some of my students are already have covered this entire thing. Can you just tell me what is the reason of this bandings? Oshik, can I expect it from you? Ah, contraction. Contraction. What are you talking about? Con- Why you are not following the class? Ah, this this bandings which are present, the banding which are present in the microfibril, what is the function of those bandings? That was my question. Good. Yeah, very good. Now tell me that how we get that banding. Like if I ask you, just tell me what is the reason of this banding. आज की हाँ. Function of these bandings. Okay. So these are the myofibrils. These are the myofibrils inside of the myofibrils. So now we will be okay. She is not able to attend any classes. Okay. Okay. I'm clearing the board, and let us talk about the detailed structures about myofibril. Okay. So next, what is myofibril? Now, after I asked how these bandings are created, you have to say, you have to answer me that how these bandings are created. Just give a hint. Myofibril and sarcomere. Myofibril and sarcomere. Today I can't see any of the boards, any of the cameras on. Give a heading myofibril and sarcomere. Akash, mute yourself.
बोलो ना
Akash, mute yourself.
কিসের ম্যাচ পাবো তারা কি বলে সেটাকে ক্লাব
কি ক্লাব
Akash, mute yourself.
ক্লাবের ম্যাচ পাবো মানে তোর মন মর্জি যেখানে একটা ক্লাব চুজ কর সেটা ম্যাচ লাগা বুঝছো কোন ক্লাব নেবে
Akash, mute yourself. Okay, thank you. I have done only.
yes ma'am
Mute yourself. I think you were talking to someone else, and that was completely audible to us. Now it's fine. Okay, let's give a heading, that is myofibril and sarcomere. Myofibril and sarcomere. So next, myofibrils and sarcomere. Myofibrils and sarcomere. So first, let us start. We will see a structure of a myofibril. See, this was actually, let's make it a little bit bigger. This was a myofiber. This was a myofiber. Inside of a myofiber was the myofibrils. So the myofibrils have compartments like this. They are having compartments like this. Suppose this is a myofibril, or this cigarette-like structure is called as myofibrils, and this compartment-like compartmental structures are called, it is just like a train, as the trains are having bogies. Okay, so these are the bogies, or this compartment, this small up to from this to this is a bogie of the, or is a compartment of the microfibril, and this is known as sarcomere. This is known as sarcomere. 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 Okay. Now the lines, these lines which are creating all the bogies, which are separating each bogie from another, these lines are called as Z lines. These lines are called as Z lines. Okay. So again I am repeating, this is a myofiber, this is a myofiber. Inside of the myofiber, this is a myofibril. And inside of that myofibril, Anisha, mute yourself. Inside of the myofibril, there are some compartments, small bogies or compartments, which are known as sarcomere. And the lines which are differentiating all these sarcomeres, the lines which are differentiating the sarcomeres from each other is called as Z line. Clear up to this part? Now, if we see the myofibril only, if we zoom in, just drag out one myofibril only, what we will see? What we will see? We will see that, suppose this is a myofibril. Okay, this is a myofibril. Now let us see a few sarcomeres. Suppose this is one sarcomere, this is another sarcomere, and this is another sarcomere. Okay, so these three are the sarcomeres. These three are the sarcomeres. Sarcomeres means this compartment, one compartment from here to here. This compartment is called as one sarcomere. This is one Z line. This is one Z line. This is one Z line. Or the gap between two Z lines are called as sarcomere. The gap show mallo left. Okay. The gap between two Z lines are called as sarcomere. Now comes the major part. The major part. Okay. This Z line is having two type of filaments. Okay, it is having two type of filaments. One is called as actin, one is called as myosin. One is the thin filament which I am drawing with green, and one is thick filament which we will draw next. So in the Z line is working as the vertebra for the this green colored line or green colored line. So it is having this filament is known as writing it down, thin filament, thin filament, which is made up of actin protein, ACTIN, actin protein. Okay. Now, in between these two actin protein, one very thick filament is present. In between these two actin protein, one very thick protein or filament is present, which is known as thick filament. This filament is known as, again here also, okay. So this filament is known as thick filament. Which is made up of myosin protein. Up to this you must be clear. So inside of the microfibrils, small compartments are present. These small compartments we called as sarcomere. In the sarcomere, there are two lines. The sarcomere is guarded by two Z lines, and the division of sarcomeres are these Z lines. First, with these Z lines, this green color filaments are present. These green color filaments are called as thin filament. These green color filaments are called as thin filament. Now, next, it is made up of actin protein. It is a protein. It is made up of actin protein, and it is also connected with some thick filament. These are the thick filament in the between of this. Again, this has overlapped little bit of the area of thin filament. This is made up of myosin protein. Myosin protein. Myosin protein. Up to this part clear or not? First tell me. Because again from this base we have to start many things. We have to understand a number of things. So this is the actin. This is the thin filament which is attached to the Z line. This is the thin filament again which is attached to the Z line. Again, if it is a Z line, some more filaments will be attached to this Z lines. Okay. So this is the main concept. Now there are multiple regions. First you write down this. Anyways, I have very less time left. Okay, done. Okay. So today we will be done up to this part only. From next day, again we will start.
Ma'am,
Yes, Anisha.
ma'am, this thin filaments means this actin proteins are connected with myosin proteins.
Okay, no, they are not connected with my actin proteins and myosin proteins. These two are not connected. Rather, they are overlapped. Just think, this is my actin protein and this is my Z line. Am I properly visible? Wait. This is the actin protein and this is the Z line in between that. So myosin proteins are little bit overlapped with actin protein, but they are not connected. Okay, they are not connected. You will come to know how they are responsible in the contraction and relaxation, but they are not connected. If this is the Z line, if this is the actin protein, the myosin protein is little bit overlapped in the area of actin protein, but they are not at all connected or attached with each other. They all today though I have not got the chance to clarify all the things in a detailed way. How does actin protein and myosin protein act or what happened? From tomorrow, I'll clarify the entire thing again. I'll start from again this sarcomere concept and I'll recapitulate. I hope tomorrow I'll get 2 classes and tomorrow or day after tomorrow this locomotion chapter will be finished. Okay, because I'm sending you the syllabus of biology. You people just go through once. Okay, your syllabus in the syllabus, just I have sent that in your group. In the locomotion chapter, you people are not having bones and all this structure ossifications, you are not having. So only skeletal muscles are there, then contractile proteins, that is actin and myosin, and the muscle contraction, that means how the muscle is contracted. Only these 3 things are there in locomotion. So we will be done with locomotion within before Saraswati Puja, within tomorrow or day after tomorrow. Next of the next of the locomotion, I'll most probably start with respiration or excretion. Okay, respiration and excretion, these 2 things I'll start. And once I am done with human physiology, I'll start plant respiration and plant growth. These two